Hello, sir.
Yes.
Sir, we have made you host, so you can share it now. You can just see once.
Okay. So is it visible now?
Yes, we can see your slide. Yes, it's visible, sir.
Okay, good.
Hello, sir.
Yeah,
Yes, it's
how
visible.
about how about the voice? Is it clear? The voice?
Yes, sir.
Fine. Okay. Okay, good. Visible now?
Yes, we can see your slide. Yes, it's visible, sir.
Okay. How about the voice? Yeah. Voice?
Yes, sir.
Fine. Okay.
Yes, we can see. আচ্ছা ওনাকে বলতে হবে আমার ওটা দিয়ে সাইডটা যেটা আমি ব্যাকগ্রাউন্ডে রাখবো শেয়ার করবো কোথায়? এটা তো এটা এবার আমি শেয়ার করবো ওনাকে তো তাহলে শেয়ার করতে হবে না আমি করতে পারবো একসাথে না আপনি তো পোস্টার ওনাকে দিবেন ডাউনলোড হবে আপনার কাছে
Sir, who are the participants mostly?
Sir, all that is teachers because it's a faculty development program. So
Right.
all faculty members under BPUT and pharmacy, Odisha. So they will be.
Okay.
Sir, for the time being, can you make me again host because I have to share one slide given by BPUT.
Okay,
So
1
after
minute.
I complete my talk, yeah, then I will hand over again to you.
Let me see how to do that. Okay. I think I should write resume share. Where we should new share, then multiple participant advanced sharing. Where I should click, there is a icon called resume share. Should I click that one or new
Yes,
share?
sir. That is on CPS mode, coordinator is given. So there you do the right click, mode, you go to more there.
More.
Then you can. It's done, sir. We have done it, sir. We have done it, sir. No problem.
Okay, great. Okay, fine.
Now share screen. Yeah. Side view. আসিফ স্যার জয়েন করেন Hello, good morning, sir. People what says okay. Make post. Post. Hello, Arsidas sir. Sir is mute আছে।
Hello. Hello.
Hello, good morning, sir.
I am audible.
Yes,
Okay, very good.
sir. Our that is speaker sir is also there. Senapati sir.
Okay. 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 Namaskar, sir.
Namaskar, sir. Good morning, sir.
Namaskar. Thank you very much, sir. Thank you very much,
Thank you, sir.
sir. Kaushik,
Sir,
what is the correct time by your watch?
sir, sharp by 11 o'clock we'll start.
No.
And,
What is the right time now? Is it 10?
now it's time for
9:45.
it's 49, sir. 10:49.
Okay. 10:49, 50. Okay.
So after 11 minutes we'll start, sir.
Okay. Wait, I'm there.
Hello. Hello.
Hello, Arsidas sir.
ଆଉ ଦି ତିନି ମିନିଟ ପାର୍ଟିସିପେଣ୍ଟ ବହୁତ କମ ଅଛନ୍ତି।
Hello, sir.
ପାର୍ଟିସିପେଣ୍ଟ ବହୁତ କମ ଅଛନ୍ତି।
Sir, I can't hear you, sir.
Hello. Am I not audible?
Hello.
Hello.
Hello, sir. Sir, can you hear me?
Yes, yes.
It's very low, sir. Volume is very low, sir.
Hello.
Hello, sir. Your voice is so low.
Am I audible? Hello. Hello. Hello.
Hello, sir.
Am I audible? Hello.
Hello, sir. Can you hear me?
I am able to hear you.
Your voice is so low, sir.
Hello.
Sir, your voice is so low.
I have net. I have in on only. Hello.
Sir, without earphone. Sir, please talk to me. Hello. Hello, sir.
Okay. Hello. Hello.
Yes, sir. Sir, I think without earphone, it will be good. Start. Hello, sir.
Hello.
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 Yes, yes.
Is it audible?
Hello, sir.
Audible. Am I audible?
Sir, now I can hear you little voice.
But my speaker is full. I am.
Hello.
I am in unmute mode only. What to do? Tell me. What can I do now?
Gaurav is calling you, sir.
Hello.
Hello, sir.
Hello.
Yes, sir.
Am I audible now?
Yes, we can hear you, sir, but it's very little voice.
So what can I do?
Sir, you can see that if your volume is okay. Phone volume. You check your phone volume, sir.
Phone volume is okay.
Phone volume is full, sir.
phone volume is full.
Shivaji sir, can you hear RCS sir? Shivaji sir,
The voice is very low.
sir's voice is very low.
So what can I do? You tell me.
Sir, check once if there are any adjustment knobs in your.
Or sir, you can do one thing. You can log out once and again please log in. We'll see.
Hello. Hello. Hello.
Sir, can you?
Hello. Is
We
it-
can hear you, but the volume is very low, sir.
So tell me what to do.
Hello.
Tell me what to do.
We can call on the phone.
Sir, is there any volume adjustment knob in your earphone? You can try adjust that one.
Uh-huh. But I am not using your phone now.
Without earphone also, it's not audible, sir.
Okay, let me try once.
What to do? Ball kichu.
So,
It's 11:11. To hear your notifications, follow the notification on your phone to turn on notification access. It's 11:12.
Hello.
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 ଟିକେ ବାହାରିକି ଆଉ ଥରେ ଚେଷ୍ଟା କରୁଛି।
Hello, good morning, Dr. Mahendra Kumar Panigrahi, sir.
Good morning, sir.
Okay, so where are you sitting on? Just below a tree somewhere?
Yes, it is Gurukula.
Okay, fine.
Wherever we are, we have to join the program. That is
Okay.
the top priority.
Okay.
Shortly, we will be starting the program. Just within 2 minutes, we'll be starting the program.
Yes, most welcome.
Jen.
Hello,
Yes, sir. Hello.
is it audible?
Audible, sir. Transparent. Very good, sir.
Full voice.
Full voice.
Okay, thank God. Okay.
Yes.
Okay, you can start now.
So let's start.
Yes.
Yeah. Okay, sir. Let's start the recording and we'll start. Okay. Namaste and good afternoon to you all. I am Kaushik Shakar, Associate Professor and Coordinator of BPUT NCFDT Pharmacy. Dear colleagues, on behalf of the College of Pharmaceutical Sciences, Mohuda Berhampur, I welcome all the pharmacy professionals to this signature event of the second BPUT Online Faculty Development Program 2022. The program has already shaped up to be excellent, and the cross-platform talk is indeed outstanding. The COVID-19 pandemic is an unprecedented tragedy in the history of humanity that has left us with grief and sorrow. Several measures were taken by the government to discover effective vaccines and small molecule inhibitors as antiviral drugs. One such initiative is Drug Discovery Hackathon 2020. It's called DDH 2020, where I participated as an individual drug designer. The platform welcomes all those who wish to join the open source drug discovery hackathon in COVID-19. DDH 2020 is a joint initiative of AICTE, CSIR, and is supported by the Office of Principal Scientific Advisor, Government of India, NC, and MyGov. This DDH 2020's vision and mission are to establish an open innovation model for in silico drug discovery against the COVID-19 virus, and will cover the various processes in drug discovery, including in silico screening of molecules, lead optimization, and identification of druggable non-toxic targets. The targets, tools, or lead molecules identified through the process of this DDH 2020 will be further taken forward for synthesis, followed by subsequent steps in a routine drug discovery program. Now, given the complex nature of SARS-CoV-2 infections that are still being understood, collective intelligence must be applied to address this global pandemic, especially in identifying drugs against COVID-19. I think globally, several scientific breakthroughs must be shared in the form of reports and discussed to accelerate the solution to the pandemic. Now, biological evolution, both in vitro and in vivo, is significant and is considered the first step towards the drug discovery cycle. Now, building on the success of previous meetings, the theme for today's seminar is animal models for SARS-CoV-2 infection. We have with us an eminent scientist, Dr. Shanti Bhushan Senapati, who holds a position in the Institute of Life Sciences, Bhubaneswar, as Scientist E. In this regard, I would like to request the Honorable Vice Principal, Mr. R.C. Das Sir, to introduce our distinguished guest speaker to the forum. Over to you, sir. I would also like to request all the members to put their microphone on mute mode, and we will not disturb our speaker while he will be on talk. And if we have any queries or doubts or any questions, so we will write it in the chat box, or it is better to keep with us, and after our the speaker completes his talk, then we will have a discussion time for 10 to 15 minutes where we will put our questions from every corner and we can again discuss with our speaker. So over to you, sir. Arshida sir, please.
Thank you. Thank you, Kaushik. Am I audible? Hello.
Yes, sir. You're audible, sir.
Good morning, esteemed BPUT officials, principals, and faculty members. I, on behalf of BPUT Odisha and College of Pharmaceutical Sciences, Berhampur, Nodal Center Faculty Development Training Program, have the honor to welcome you all to this second Faculty Development Training Program in Pharmacy of BPUT on the topic Animal Models for SARS-CoV-2 Infection. I am very much thankful to our Honorable Vice Chancellor, Professor Dr. Chittaranjan Tripathi sir, for introducing the Faculty Development Training Program and Career Counseling and Guidance Programs and Industry Institute Interaction Programs for Pharmacy faculties and students with the objective of improving standards and qualities of the faculties, students, and institutes with the World Bank funding. I am thankful to the ex-Director Curriculum, Dr. Shivkumar Mishra sir, and the present Director Curriculum in charge, B.B. Mukherjee sir, and their team for processing, assisting, and facilitating for the conduct of the program in time. I am thankful to our Dean, Professor Dr. S.K. Mahapatra sir, and the principals of the pharmacy colleges of the state for their full support And meanwhile, I think already 6 such programs are conducted. And today, 7th and 8th programs are going to be conducted by College of Pharmaceutical Sciences, Mauda and TPC Barpali. And we have already conducted 3 faculty development programs. And this is the 4th faculty development program of BPUT and 2nd faculty development program of College of Pharmaceutical Sciences. I am also thankful to our principal, Dr. Subrat Sen sir, who has personally taken care and encouraged us for successful conduct of this program. At last, I must thank our Nodal Cell Coordinator, Kaushik Sarkar and team members, Subhasis Misra and Gaurav Saha, who facilitate and manage the entire program. Once again, I extend my hearty welcome to you all to this scientific faculty development and training program. Now, on behalf of you all, I welcome Dr. Santibhushan Senapati, Scientist E, ILS, Bhubaneswar, the esteemed speaker of this faculty development and training program. Dr. Santibhushan Senapati sir, please. I am very much thankful to you, Dr. Senapati sir, for accepting our request and accommodating this program in your busy schedule, that too in a Sunday. Let me introduce Dr. Senapati sir in brief. Dr. Santibhushan Senapati is a Bachelor in Veterinary Science and Animal Husbandry from UAT and PhD in Biochemistry and Molecular Biology. And at present, he is Scientist E in ILS Bhubaneswar, Odisha. Before giving the brief introduction of Santibhushan sir, I would like to tell little about ILS. We all know the contributions of Institute of Life Sciences, Bhubaneswar, in fighting against COVID-19 pandemic. On April 14, 2020, ILS, with approval of ICMR, started testing COVID-19 with just 44 samples in their BSL-3 lab. And by 16th May, they reached the testing about 1,500 samples per day. Why we all confined ourselves at home? Dr. Santibhushan Senapati, one among the team under whose leadership the ILS started working day and night tirelessly for COVID-19 testing. ILS team played a major role in containing the COVID-19 pandemic. Also, immediately, ILS focused on development of COVID-19 vaccine while maintaining the highest standards and safety by engaging their virologists and molecular biologists. So this is ILS in brief. Now, coming to the brief biodata of Dr. Senapati sir, he worked from 2000 to 2005 as a veterinary officer in government of Odisha. Then he switched over to the challenging career to the research field. And 2005 to August 2005 to April 2010, he joined as a research assistant in University of Nebraska, Omaha, USA. And from August 2010 to February 2012, he joined Ohio State University, Columbus, Ohio, USA, with postdoctoral research as a postdoctoral researcher. Then April 2012 to September 2015, he came back to Institute of Life Sciences and joined as a Ramalinga Swami Fellow. And September 2015 to December 2018, he joined as Scientist D in ILS Bhubaneswar. And from January 2019 till date, he is working as Scientist E in Institute of Life Sciences Bhubaneswar. And he has completed 2 projects. That is one DBT project as a principal investigator, functional role of MAGEA3 cancer test antigen in pancreatic cancer, worth 42 lakhs. And as co-PI, funded by Science and Engineering Research Board, implication of bacteria-derived human defense in burn injury and wound healing, worth like 45 lakhs. And at present, three major projects is working, and that is the functional consequences of cancer testis antigen ATAD2 in pancreatic cancer, funded by DBT as co-PI, worth like 25 lakhs, and to characterize the exosomes secreted by chemotherapy exposed pancreatic cancer associated fibroblast cells and investigate its role in chemo resistance of cancer cells, funded by Science and Engineering Research Board as principal investigator, worth 25 lakhs. And the major project now is handling establishment of a platform for experimental animal model to carry out preclinical evaluation candidate antiviral vaccines against SARS-CoV-2, funded by DBT BIRAC as principal investigator, and its worth is 10 crore 61 lakhs 20,000. So such a major project he is handling, and he has already produced 3 PhDs, and currently 7 students are working for their PhDs, and postdoctoral training completed 2, and currently 1 is continuing. And during last 5 years, he has contributions of 21 research publications from their laboratories, and he has a major contribution for the life sciences. And he is a young dynamic scientist, and I'm very glad to know that his date of birth is 28th May 1978, only yesterday only. So I have the opportunity that on behalf of members present here, I extend a happy belated birthday to you, sir. Let the Almighty fill your life with all happiness. Almighty fill life with all happiness and blessings, you to achieve new heights and goals in your life. I hope that this deliberation and interaction will more fruitful and successful, and I hope for a interesting question and answer session that is the vital part of a faculty development and training program. Now I request Dr. Senapati sir to start his lecture. Sure. Senapati sir, please.
Namaskar, sir. Sir, thank you so much for this generous introduction, and good morning to all. And I'm thankful to the organizing committee members for giving me this opportunity to be part of this training program. And thank you, sir. And so what we'll do, sir, like I'll request to allow me to share my screen, and then after we can go ahead. Am I audible, sir? Hello?
Yes, sir, you are absolutely audible.
Okay.
Hello? Yes, sir, you are the host now.
Okay. Yeah. So once again, thank you very much for attending this talk. And so what we'll do, actually, I realized that we have around 1.5 hours with us. So as all of you are well aware of this COVID-19 and also some aspect of the ongoing related studies in this field. So what we'll do, 1 hour I'll spend just to discuss whatever current understanding that we have related to different animal models that are being used in COVID-19 research. And the last half an hour we can have for question and answer sessions. As Sir rightly mentioned, I look forward that section more eagerly so that we can share some of our thoughts and clarify each other doubts. So basically, as when Sir was talking about the activities of my lab, you must have realized that I am actually currently a cancer biologist. Although my basic training is on animal science as a veterinarian. So after that veterinarian, I entered into molecular biology study, and most of my studies are related to cancer biology aspect. But when this pandemic came, that time, as I have an inclination towards animal models and we do animal work in different aspects of cancer, so I thought, why not to use our expertise and contribute some aspect of this COVID-19 research. So what I'll do, I'll also, initially, I'll take you through some of the general aspects of COVID-19 animal models. Then the last part, I will mostly discuss about what we are doing here at ILS, what we have done. And most importantly, as Sir just mentioned, that we have a major project going on here to establish a animal content biosafety level 3 facility for COVID-19 research. And especially BIRAC has funded this project to run this facility on a service mode, fee for service mode. So I think that will be very interesting to share with you all in future. If any of you will be interested to carry some of the studies, we can definitely facilitate that here. Or also some of during my discussion, if you will pay attention, some of the things might also help many of you to establish such kind of facility at your institute, because nowadays government is promoting to establish BSL-3 and ABSL-3 facility at different part of India. So in future also some of these ideas might help. So as all of you know, like whether it is COVID-19 research or any field of biology, we can just go with just one aspect of that particular field. Like when we'll take, so for an example, when we talk about COVID-19 research, is such a huge area, like starting from understanding the basic aspect of the pathogen itself. Like when we talk about COVID-19 research, first thing is to understand what is this pathogen is about. It's a virus or bacteria. Then people went through and understand that, okay, it's a viral disease. Then people got interested to know its genome. When we say that its genomic characteristics and proteomic characteristics, it itself is a huge area of research. Then understanding its replication process, mutagenesis process. Similarly, the next field is, once you understand, okay, this is the pathogen, what is the pathogenesis process, how it gets spread, like what cells, what kind of animals get infected, and what is the mode of transmission, right? So whether it transmits through nosocomial route or fecal route or contact. Then you have to understand also how, when we say pathogenesis, it itself is also a huge field, like what kind of cells really get infected, why there is a specificity for particular cell, not other cells. So it's an ongoing and evolving area. And then another major study is like once you understand the genomics and a little bit about pathogenesis process of a pathogen, the next question is how to content it, how to or the treat, what are the preventive measures that can be taken, what are the treatment modalities that can be adopted. So all these things are important when we talk about any infectious disease. So to do all these things, we definitely need some of the research tools, models, and or reagents like. So in this regard, when we say that experimental things that we conduct related to infectious disease, model systems are very important, right? So, and when we talk about model system, always the models depend on the question. All models have their limitations and potential. So, so most important thing is what is our question. So based on the question, we have to pick the model. Like some of the studies, for some of the studies, the cell culture model is good enough, right? Suppose where isolation of virus. So for isolation of virus, there are standard techniques. You must be knowing some cells like vero cells, which are permissive to different virus, which they get infected due to their genetic makeup for different viruses. So cell culture model is good enough for isolating the virus, propagating them. Like that, if we go a little bit more organized model, which mimics the in vivo system better. Nowadays, people are using organoid models, which are miniature of different organs of human being or any animals. So organoid models are heterogeneous cell population. So they give little bit better information about what kind of cells get infected. And so it is better to understand the the pathogenesis that we may miss in normal cell culture model. Then comes the animal model. So animal model in clinical or biological research is very important, and I am sure you all of you are well aware of it, being in pharmacy or pharmaceutical background. And without this, probably many things that we have today, many medicines or intervention that we have today may not have possible. And when we say about animal models, again, it is the which model to pick. Like the most important thing, does that model has any clinical relevance or not? So how much it mimic the human scenarios, right? And again comes the question, like for certain questions, one animal model might be good, for other questions it may not. So all these things are very important before picking one animal model for a study. Understanding the model or the animal first, then understanding what is the strength and weakness of it, and correlate it to what question we are addressing, right? So when this SARS-CoV-2 got identified, so as many of you know, even still the debate is going on, like what are the origin of this SARS-CoV-2? Like many people predicted that, or proposed that it has been originated from bats, because the SARS-CoV-2 genome, when got sequenced, people realized that it has many similarity with earlier SARS virus, which was identified to be originated from bats, right? So they thought like probably this SARS-CoV-2, also this new variant of COVID virus, is also from bat, but it is still under debate. There is no clear-cut understanding. And people say that then pangolin is probably one intermediate host through which it has come to human being. But still, like all these are still speculations only, based on the earlier understanding that SARS-CoV-2 can infect these animals. And also, like they particularly, all these assumptions revolved around one point, that these animals have the target protein, the ACE2 protein, the binding protein for SARS-CoV-2, which has very similarity or binding affinity for the spike protein. So all these in silico studies actually modulated our thoughts to believe that probably it has come through these animals. So, but definitely these informations that animals are the transmitting host for many of the infectious diseases that we encounter in human beings. So it is nothing something new that people predicted, as history says that the zoonotic diseases or even the involvement of animal in human infectious disease is paramount. So very quickly we start believing that there must be many other animals, not only human beings, who may get infected with this virus, right? So that immediately, we all are kind of a little bit selfish, definitely. Our intention to understand which animals get infected with SARS-CoV-2 was not to protect these animals, definitely at the initial stage. We are more interested to know like which animals we can take as model to understand this virus better, or can identify some therapeutic or preventive measures, so that first human will definitely get protected, then all these animals can also get protected. So that is the reality we have to accept. Like animal models have been always helpful to human beings, but on return, what we get out of this animal model research also in return are equally helpful to the animals too. So as you can see, like all these animals, animal models, like they are important for to understand basic things, like what we have seen in case of human patients, like young people are less susceptible, or children are less susceptible to this infection compared to older one. So that you can ask the same basic question: does it happens in animals? You can ask even bigger question, like what are the route of transmission, like as we discussed, like whatever associated field related to this infection, all questions can be asked. Then importantly, to find out the therapeutics, right? We have to go through preclinical animal models to understand the toxicity, then efficacy, all these things. You all of you are well up and well versed. So all these things are very essential where preclinical animal models play a role. And importantly, we now as the pandemic has reduced, like the scariness after, fortunately, all this vaccine development and all these things have made us to control this up to a certain extent. And now the research field is moving towards understanding the role of after infection, long-term sequelae of this infection, right? And what is the long-term effect of COVID-19 infection, and also importantly, why and how the comorbidities play a role in aggravating this the manifestation of this disease, right? So now these are also ongoing and important topics where animal models are playing a role to address that. So as all of you know, like this particularly in human being, what we noticed, like all people didn't manifest the disease similarly, right? So some are asymptomatic, some are symptomatic, which are either mild or severe. So it manifested in a different way, and some things we understand, and actually most of the things we don't understand why it happened like that. And and mostly those people who suffer from severe infection, or who got hospitalized, or some who are under intensive care unit, So in most of them, the problem was like a longer persistence of the virus in the lung, and importantly, the uncontrolled host response. What we say that the aberrant immune reaction or the hyperactivation of immune system, which didn't get controlled by the host system, lead to more damage, not only in the lung tissue, also in other vital organs and like kidney, heart, and other places. So, and which lead to multi-organ failure. And most of the people who have comorbidities actually added more difficulties in this scenario. So, actually, when we talk about animal models, the good thing with animal models are like they are controlled system. Like you, whatever animals, suppose 5 animals, 10 animals, whichever animal you take for your study, you know that what is their initial health status, and most of them are of similar type, you know, like so that the end result that we see, there will be less variability. But at the same, but in case of human being, we see a lot of heterogeneous population, age, sex, the genetic makeup, all these things add to that. So the good thing with animal model is it is homogeneous and controlled one, but the bad thing is it doesn't mimic the heterogeneity that we see in real clinical scenario, right? So far, whatever animals are being used in COVID-19 research that we'll see one by one, most of them actually, they recapitulate the milder or moderate form of COVID-19 disease. And almost none of them actually recapitulate the complex scenario that we see in human context, like particularly the comorbidities. When we talk about the comorbidities implication in COVID-19, that part we miss in animal research study. But at the same time, like the good thing with animal, although the models are the models show moderate or less severe form of COVID-19, but they are good enough to carry out studies like vaccine research, antiviral studies, and up to some extent the pathogenesis studies also. So we miss so far, we don't have very good models of severe COVID-19 that we see in human beings, but whatever we have, we'll see one by one. They have also importance so far in this particular field. So when people can initially characterize this virus and understood that it is a COVID virus, and immediately the scientific community got attracted to a protein that is called AC2 protein, right? The AC2 protein actually is a already known receptor or entry point molecule for the earlier COVID virus, SARS virus, around 2005, 2006. People had already knowledge about that SARS virus bind to AC2 protein and enter into the host cells. And consequently, it replicates within the cell and causes the pathology. So immediately people thought about to use a AC2 transgenic mice. So fortunately, for a such study, people had already generated these mice earlier. For a long time, these mice, people didn't pay attention to these mice because the SARS research funding was going down, and the person who generated this genetic engineered mouse, which is expressing a human AC2 protein under a promoter of K18, like a epithelial specific promoter, which will express the human AC2 protein in different organs, particularly including the lung. So these mice were actually with Jackson Laboratory, which is located at US, and they have consortium of different genetically modified mice, which they commercially provide to users. Okay, so immediately, so these mice were there already, and so it was very useful for the scientific community to revive these mice. In Jackson, immediately they had frozen germplasm, sperm, and ova, all these things. So immediately they took out all those stuff and revived this mice colony and expanded them and started providing to research community. We also at ILS got this from Jackson. So we have also this mice. So these mice actually, but as per the prediction, it also turned out true that they are highly susceptible for SARS-CoV-2 infection. And that itself again proved that this AC2 molecule is probably a major mediator for SARS-CoV-2 pathogenesis. Although in recent studies, we have realized that it is not the sole molecule through which SARS-CoV-2 enters into host cell, but definitely it is one of the major molecules. So in this mice, it has also, as I mentioned, all models have strength and weakness. So the good part with mice model is like they are smaller, they are easy to breed and expand their number, and also the maintenance of these mice is less. So just for your information, like when Jackson provided these mice to the users, there was a strict instruction that these mice should be handled under BSL-2 facility minimum, because like if we are taking these mice and infecting them in an animal biosafety level 3 facility, that is okay. But even most of the time when we get mice breeding and all this thing doesn't happen in biosafety facility, only experiment happen in biosafety facility. So before to that, you have to keep these animals, breed them, right? So all these things in normal animal house is not recommended, because as there is a chance of transmission of virus from the animal to human, and human, more importantly, human to animals also. So as more, if any of the handler is having COVID infection, there is a high chance that it will spread to these mice. So there was a strict instruction that all these mice should be handled minimum in biosafety level 2 facility. And the handlers should be oriented. They should be also equipped with PPE so that we can avoid any spreading from each other. So these mice are highly susceptible. There is no problem in breeding them, handling them. And so they also manifest definitely in COVID research, the first organ that draws our attention is lung. So whatever model, whichever model you use, if lung pathology doesn't mimic what we see in human, then it has very less importance. So when people started exploring different animals that can get infected with this virus, so first thing they always characterize is what is happening to the lung. So in these mice, people saw that yes, we see some kind of lung damage like the interstitial pneumonia, also vasculitis was also present. And this animal model also, the severity of the disease depends on the virus inoculation, the number of virus particles that we infect or give through intranasal route to these animals. So more than 10 to the 5 clock-forming unit turned out to be lethal to this animal. So which is also a good model. When people do animal studies, some of you must be realizing that lethal model means at a particular dose, you know that after 5 days or 6 days, these animals will die. So you have a clear-cut black and white readout that animal survived or animal died. So in that case, things get little easy. Suppose you are doing an antiviral study, you know that whether if your drug is really good, you will see that the animal are surviving. If it is not working, you will see the animals died after 5 days. So lethal model, the good part is you know the end point very clearly. If it is not non-lethal, it has also importance, but the point is it will be little bit relative. Means the animals, if animals are not dying, you have to sacrifice the animal, then quantify the viral load through different methods like qPCR method, plug funding unit. Then you have to also do histopathology of lung and other organs to score what is the level of damage, lung pathology score, and all these things will be done. So it will be a little bit relative, but it's not like they don't have importance, because if it is a non-lethal model, it is good for vaccine study. So if the animal is not surviving, then what long-term study we can do? We cannot do. We can do any study like suppose you injected the vaccine candidate now and want to check whether challenge with virus will work or not. So you need a little bit longer observation time. So lethal models are not good for vaccine studies. So the good part with this mice model is you can control your virus load that you are, or challenge load, and accordingly you can design your study. But the negative point with this model is, like, when you generate a genetically engineered mice, you don't have a control like in which tissue it will get. Although the promoter is specific for epithelial cells, so in addition, this ACE2 protein, in addition to lung, also get expressed in other organs like brain and mostly in brain cells, because in human, we do not see brain involvement that much. Although some studies, observational studies are suggesting that brain cells might be getting infected, but that is not the common one. Very rare incidents you can probably find where COVID virus or SARS-CoV-2 virus infects the neuronal cells. But in ACE2 transgenic mice, as the transgene gets expressed also in neuronal cell, so these mice also show neuroinvasion and brain involvement more compared to what we see in human. So some of the mice that die after infection, people believe that it may not be due to only involvement of lung, rather it is due to involvement of brain also. So that is a negative point of this model. Like as I said, like all depends on what is your question. If an investigator has a question like what COVID virus does to neuronal cells, they may also find some potential in this unexpected phenomenon. But those people who try to understand real pathogenesis and organ spreading of this virus in a clinical context, for them it may be a negative point. So these are the things associated with this model. When people were looking for this mice, because as I mentioned, these mice were not commercially readily available. So it took around 5-6 months to Jackson to expand the colony and provide worldwide to carry out research with these mice. That time people had already started infecting the normal mice. As you know, normal mice are also of different types, of different strains, black cyst mice, bald cyst mice. So all these have also some genetic difference. People started infecting those normal mice also, and they realized that these mice, the wild type mice, are not permissive to SARS-CoV-2. Means they, if you infect them, they within 1-2 days, virus is getting cleared. The immune system is robust to contain the infection. So probably people believe that the wild type mice, the mouse ACE2 protein probably doesn't have affinity towards the spike protein. So that's why people thought that either you have to go for humanized AC2 transgenic mice. One mice I already discussed, which is already available commercially, and genetically that AC2 has been incorporated into the genome of that mice. So another quick method people adopted, where people like adenovirus expressing this human AC2 protein, like virus particle, they commercially, many of you might be using to overexpress certain protein by lentiviral system or adenovirus system in cell culture. Likewise, they use this adenovirus system, and where the human AC2 protein will be expressed to whichever cell this virus will infect. Means what they did, they intranasally inoculated this virus particle carrying this human AC2 gene, and when they infected, some of the cells of the lung will take up this virus and they will start expressing this protein. So then second challenge they will give with the SARS-CoV-2 virus. Whichever cells are expressing this AC2 protein are expected to get infected with SARS-CoV-2 virus, because the human AC2 will be on the surface of those cells. So they succeeded. They use this SARS-CoV-2 viral, so here, okay, so this second point, you can see the human AC2 transduced mouse. So transduction means when you give a viral particle and the virus infect the cell and start expressing your desire of protein, right? So this is the AC2 transduced mouse, means where the people use this SARS-CoV-2 infected virus particle to express human AC2, and then they became permissive to SARS-CoV-2. But it has some limitations also, because the earlier virus that will use to express human AC2 by itself also will cause some of the inflammation in the lung, and which may actually make it difficult to interpret the lung damage that you saw is really due to the SARS-CoV infection or the adenovirus system that has been used to overexpress this protein. But people succeeded in this method too. Then another interesting thing which came down the line is mouse adapted SARS-CoV-2 strain. So what happened? Like when people got expertise in infecting mice or other animals, and they have understanding like how long these mice can survive, all these things. And when they understood that the wild type mice, they are not susceptible to this infection. So what they did, they infected wild type mice with SARS-CoV-2 virus, and after certain days, they harvested the lung tissue. Again, they cultured those viruses from the lung tissue. Okay, so the lung infected animal, they sacrificed, they took the lung tissue. Then from the supernatant of the lung tissue homogenate, they again infected in culture some of the cells, vero cells particularly, which people use to propagate virus. Then after passing in vitro for 1-2 passes, they reinfected those virus again in another set of animal. Means again and again you are passaging, like in cell culture we do cell passage. In animal, they went on passaging the same virus again and again. So with a belief that some of the virus, as this virus has an intrinsic property to mutate it itself rapidly, so it may happen that some of the virus will find naturally a mutant which will help them to survive in wild type mice, and it happened. So after 5-6 of passes, they found that some of the new variants that emerged during this process now can even infect wild type mice. So naturally, some of the mutant variants got selected, which has ability to bypass the mouse immune system and infect mice also. So these are called as mouse adapted SARS-CoV-2 strain. And particularly when we do this kind of study, we have to be extremely careful because we are selecting a more aggressive variant, but whether it is aggressive variant for human being, we don't know, but at least for mice, it is an aggressive one. So it may happen that by chance this variant will escape from a laboratory, it may spread through wild mice or rodents like that more severe. So when we go for this kind of modified variants or select some more aggressive variants, so that is one thing ethically we have to be really very careful about it. So now comes the animal model, the hamster model, and one of my favorite model, because my lab was working with a hamster model of pancreatic cancer since last 10 years here at ILS. So we have hamster breeding going on at our animal house. So that time when this pandemic, early part of the pandemic, immediately it drew my attention, like when people told that hamster is a potential model, because earlier studies have shown that the SARS virus can infect hamster. And actually hamster is such an animal, like it is permissive to multiple virus, not only SARS virus. There will be around 10 to 15 virus which can easily infect hamster. So people don't have actually clear-cut idea why hamster is susceptible, which is a wild type animal, and it is immune competent. It's not like an immunocompromised animal, but still it is susceptible to many human pathogens. Okay, so and one reason people believe that probably the interferon pathways are less active due to the genetic structure of interferon related molecules. They are not that potent to eliminate this virus as we see in other animals or in human being. But that experimental, that has to be really validated or proven. But that is one thing which people suspect. So these hamsters, which are actually a little bit bigger than mice. So mice general body weight, you will see around 20 to 27 gram based on the age and other things. But hamsters, the adult hamsters of more than 3 months old or 4 months old, you will see body weight varying from 60 gram to 90 gram like that. And so, and in these animals are also, many of you might be knowing, like the females are actually bigger than the males. And so these hamsters actually, people saw that yes, they also get infected with SARS-CoV-2. And the good thing is they manifest most of the lung pathology that we see in case of humans. And this is the one of the model that we are very frequently using at ILS. And I'll discuss some of the aspect later part of my talk. So these animals, as I mentioned, they are good. They show lung pathology, that they show bronchitis, alveolitis, and also the most important feature is vasculopathy, the involvement of blood vessels, the endothelitis, the perivascular coughing or infiltration of immune cells are very clear cut in these animals. And surprisingly, these animals don't die. It's not a lethal model. Like when you harvest 4 or 5 days old infected animal's lung, you can even out from the outside grossly, you can see there is a hemorrhagic patch, lot of congestion, lung is totally inflamed. But surprisingly, the same animals, when you keep for 14 days around and you harvest the lung, you will see everything is clear. They have a capacity to recover very quickly. But that's why most of the readout that we take this model are between 4 to 5 days after infection. So this is another animal that in case of hamster, I must say that the good part is they also develop your antibodies, okay, neutralizing antibodies that what we see in human being. Means once you infect one hamster with SARS-CoV-2 and it recovers, then again, if you re-challenge it, it will not get infected. Okay, so that means that so that they have a capacity to generate antibody against SARS-CoV-2, which was essentially to know for vaccine studies. So many, like if you will go to PubMed, like so far, whatever animal studies have been done, you will see around 60% studies are done in hamster model. And then 30% you will see in mice, and 10 to maximum 10% or 15% you will see in case of monkey model or mammalian model that we'll discuss. So hamster turned out to be a major model in COVID-19 research due to two things. It's lung manifestation of the disease, and the other thing is its capacity to mount immune build immune activity against the virus for vaccine research. So these ferrets actually they are very difficult to work in lab due to their bigger size and ethical concern. So as the animal size goes high, the ethical concerns also become very tough. So and the other problem with these ferrets are like the lung pathology is not that high, that kind of distinct as we see in human one, but they also show some involvement of lungs. In case of when we say about the closeness of a model or recapitulation of the things in the context of human beings, so definitely nothing can be better than a mammalian model, which is a monkey model, okay, non-human primate model. So people tried, because when we talk about primates or monkeys, there are also different type of monkeys. So people tried different type of monkeys. You can see here rhesus monkey, then African green monkey. All these things people have tried. So first problem with the monkey model is the ethical thing. All the animal house are not allowed, or they don't have license to handle bigger mammals like monkey. Only probably in India you'll see only countable 1, 2, or 3 labs are there, or like NIV, and probably then biocontainment facility at Bhopal. These are the two what I know. There might be another 1, 2, but who are allowed actually to do monkey research here. So otherwise, most of the animal facilities don't have, including ours also, we don't have permission to carry out non-primate model experiments. Okay, so it needs definitely some higher expertise, and also most importantly, the number of animals that we can use in a study will be very limited. So you have to be really very clear about the question or the intention where you want to use these animals for the study. And there are multiple publications who have clearly shown that rhesus monkey can get infected, and they also show the lung pathologies that we see in case of humans. And importantly, they also mount the IgG level, the neutralizing antibody level, to give immunity for re-challenge study. So in case of vaccine related study, even most of the vaccines that are available nowadays in market, they must have gone through at least two models, so one hamster model and the monkey model, where to show the efficacy and the safety level of those candidates. So as you can see here, like most of the problems, like as I told, the strength is their AC2 structure, the protein AC2 molecule structure is very close to human. That's why the level of infectivity that we see in case of human, also we see in case of monkey. But there are also a lot of difficulties or bottlenecks associated with this model. So this is a compilation. This table shows a compilation of things that we see in different models. Like if you talk about upper respiratory tract involvement, probably most of the models show that. Mice, hamster, ferrets, non-human primates like monkey, all these models show that upper respiratory tract involvement. In lower respiratory tracts environment, you can see hamster and non-human primates are mostly you can see there. So in case of COVID-19, you might be knowing like some of the non-pulmonary organs, lung organs like brain, kidney, all these things people speculate or have some evidence are also getting involved. So you don't see other than lung involvement of different organs in all models. So even if you see, you don't see up to that extent. But one of the major other organ than lung is the gastrointestinal tract, right? GI tract. So because the ACT proteins also get expressed in many of the intestinal epithelial cells. And you must be knowing like diarrhea was also one of the common symptoms in COVID-19 patients. So the GI tract involvement also important for many of the investigators to evaluate, to understand the spreading of this disease, and most importantly also to evaluate different candidates that can control the GI transmission of this pathogen. So GI transmission was common like in mice and hamster, also in monkey, like people reported that this virus is also getting spread or the shedding is happening to the fecal route. But the involvement of the damage to the GI tract, infection of the epithelial cells, the necrosis or infiltration immune cells, all these things that you expect was not that robust. Like you see, when you will check the virus genome, you will see that it is present. But presence of virus genome doesn't guarantee that it is infecting your cells. Okay, even the inactive virus will have also genome detected through qPCR or any other molecular technique. But whether the cells are really getting infected or not, for that you need additional storage. And in some cases, people when they check for viral protein through techniques like confocal or immunofluorescence staining in different organs, they saw some of the evidence that some gastrointestinal epithelial cells are getting infected. But in all the models, the bottom line is the severity of enteritis was not that high. Even in human, we don't know, like because in human system, it's not possible, ethically possible or technically possible to see in COVID-19 patient really how much the enteritis is happening and which part of the gut is actually getting more affected. It is the large intestine or small intestine. So that knowledge also we don't have. But the point is, in these animals, we do see some of the intestinal involvement, but the extent of enteritis is not that high. When you talk about clinical signs like fever, nasal discharge, and all these things, only ferrets showed fever, increase in body temperature, and nasal discharge. That's why when we talk, if your question is related to nosocomial spreading, a nasal spreading of understanding the nasal spreading of this pathogen, then ferrets are really good because they also sneeze and their temperature also goes high, and so that is a good model to understand the transmission of this virus. But the nasal discharge and all these things we don't see in any other animals. We have seen like in case of we ourselves have seen that in AC2 mice, we do see in severe cases there will be a discharge. The ocular discharge will be there, the eyes will be swollen and all these things. But the nasal discharge we have not noticed, and definitely not even in hamster. Now hamsters are like they recover very fast without really much clinical manifestation, that like we don't see that kind of severity in hamster. And so yes, labor breathing up to some extent we see in hamster, but irrespective of all these models, one very typical readout of severity is the body weight loss. In case of AC2 mice, you will see that within 5 days, most of the like severely infected animals will lose around 20% of their body weight, and which is actually a point where you have to declare at human endpoint value, means you are close to dead only. Below 20% decrease in body weight, you can, after that you cannot ethically keep these animals. Okay, humanly you have to sacrifice them. So in case of hamster, the body weight loss although is there, around 10%, 12%, 50%, it is like that. They never go beyond 20% body, and gradually it will recover to normal within 14 days. But in case of AC2 mice, some of the animals actually succumb and they die. So related to lung involvement, as I have already mentioned, like a hamster is a good model where we see a lot of features of lung involvement, and definitely the non-human primate like monkey, they also show the lung involvement. Transmission we see in hamsters, ferrets, and cats. So when we talk about the immunology point of view, like because this is very important for vaccine research, two most frequently used are hamster and non-human primate like monkey, and up to some extent the mice also manifest the seroconversion. So this table shows like what are different studies so far people have already done, starting from type of studies. You will see infection and pathogenesis, therapeutics, vaccine. 1 minute. Okay. So infection, pathogenesis, vaccine studies, all these things people have tried, and one very important thing is this comorbidity study people have started doing with non-human primate model, and most importantly the pathogenesis study, vaccine candidate or antiviral candidate, people have mostly used hamster and mouse model so far, and vaccine studies have definitely the non-human primates have been used. I think this slide we can skip. So now I'll just quickly take you through what we are doing here at ILS. So when I was telling, like at the early part of the pandemic, we had the hamster already with us for different other purpose. At the same time, two of my colleagues who are the virologist, Dr. Ghulam Hussain Said and Dr. Soma Chattopadhyay, both of them actually did a good job. They isolated COVID virus from the clinical samples which are coming to ILS for diagnosis, right? So we got ethical approval, and with due approval for all the, fortunately that time we had actually BSL-3 facility at ILS. So we immediately took some of those samples and isolated virus from them. And these are all local isolates from Odisha. And gradually we went ahead, we characterized all those viruses at the genomic level. So whole genome sequencing was done. Then we confirmed that they are indeed SARS-CoV-2 virus. And then at that time, my lab and Gulab lab, we came together and thought about to establish this animal challenge model at ILS. So using this locally isolated SARS-CoV-2 virus, we infected the hamsters. And as people have already reported at different other parts of the world, we also clearly see that these animals are indeed getting infected. And we went further, actually, we collected the lung tissues of 4 days infected animal and 14 days infected animal, did a proteomic study and saw the differentially expressed protein in these 2 time points. So that already published, we have published in the SF journal. But as you can just see glimpsely, like what we see, this is the one is the normal lung, mock infected. Only intranasally we have given the PBS. This side lung is the SARS-CoV-2 infected one. You can see the way the hemorrhagic patches have clearly, or congestion are clearly visible in the surface from the outside grossly. So you can imagine the severity of lung damage in hamster. Surprisingly, everything goes away within next 4 or 5 days. By 14 days, so this is after infection, you will see this kind of things within 4 or 5 days. But if you wait for another 5 or 6 days, these things are gone. So in case of hamster, actually, they have multi-lobe lung. The right side lung is a single lobe, but the left one has around 3 lobes. So, and all of them, actually, there is no control which lobe will take the virus. It randomly happens. Sometimes you see right lobe has more congestion, the damage, sometimes left lobes are involved. So, and you see what we see under microscope. When we did the histopathology, the white patches, the balloon-like things, I don't know whether the cursor is visible or not. The balloon-like things you see here, the white patches in the mock infected one, actually the alveoli, the free space where the air actually stays. The normal lung should have this kind of patchy white areas. Okay, but you see in this side, the SARS-CoV-2 infected one, the V is the bronchus. The bronchus is full with, here you see the V and the infected V. Bronchus is full with immune cells. The high dense blue color are the polymer for nuclear cell, the immune cells. Okay, and this is a blood vessel. You see here, it has a perivascular coughing. Immune cells totally surrounding the blood vessels. Within the blood vessels, you see a lot of immune cells attached to the wall of it. So this is called endothelitis. Endothelitis, bronchitis, all these are very prominent in this hamster model. And when we go for evaluation of anti-candidates, antiviral candidates, all these things, we give a lung pathology scoring, like to compare them. So here you can see these are the different features that we look for when we score for lung pathology. The endothelitis, consolidated area in the lung, the hemorrhagic area, the necrotic bronchitis feature, all these things we take into consideration. The pathologist and sometimes I also do, we take into consideration of all these features before giving the pathology score to these ones. So this is the compilation of things that we have reported when we characterize the hamster model here at ILS. And this confocal data, this red stain things are against the virus protein. You can see that the virus protein are clearly present in the bronchi of the infected animals. And these are the mock infected where it is absent. So lung damage was there, gross congestion was there. And one very feature which already people have reported, like these surprisingly in these infected animals, the number of the epithelial cells also divide very fast. That's why they have Ki67 and the proliferation marker is very high. Probably this is a self-adaptation. The body, the host body is trying to replicate these epithelial cells to compensate the loss or damage that has happened. So this is some other study that we have already done here at ILS. I'll skip that one. And here, this is a microcity. I'll talk about a facility that we have recently established here. But before that, in that facility, so that facility was funded by BIRAC. So initially, we had already one BSL-3 facility where initial studies were conducted. All the initial establishment of the modern and all these things, we did the existing BSL-3 facility that time. Then when we showed our expertise and all these things, the BIRAC got convinced and gave us money to create another new facility exclusively for SARS-CoV-2 related studies. And importantly, as I mentioned, the condition was this facility should run on a service mode, fee for service mode, and it has to become self-sustainable. In that facility, so we got money not only for establishing the infrastructure for ABSL-3, but also to get some high-end equipments like microcity scanner. So microcity scanner, as in human, people do X-ray and microcity to radiological techniques to image the lung and estimate the lung damage or involvement of lung. So this is our own data. You can see the left side one is the hamster lung. The more black you see is better because air should look like, due to low viscosity, should look black or gray in radiological image. And this white patch actually is a heart area. So if you see here, these two black dots are the bronchi, and around the bronchi in the SARS-CoV-2 infected one, you see two white patches around. So this means these areas have got consolidated or damaged. So there is no alveoli, functional alveoli are there. The air, they have been totally filled with exuded the immune cells, edema, and also hemorrhagic area. So a lot of things also actually have made these areas devoid of air. So that's how microcity is also very, very useful for us to image. Even the good thing with microcity system is it is a live animal imaging system. You can do multiple scan at different time points to see how things are progressing with treatment or without treatment group. So this is the new facility that we have developed within 1 year time. Definitely it would have been not possible with the support of our director and definitely funding BIRAC and funding from BIRAC. So all this is, you must be realizing creating a facility is not one-man job, although I was the PI, but the whole system supported me. I'm starting from administrative, store purchase, all these people have to be in the loop to make this possible, because it was only a 1 year project where we committed that we'll expand. The backside one is the old one. This is the new facility that we expanded one exclusively for SARS-CoV-2 study. So this is from outside, you must be seeing that it's looking very simple, but actually these are now all prefabricated BSL-3 facilities. Companies are there who can develop this kind of, there are containers. So there are metal, these are metal containers. From the top, you are seeing this fiber sheets or metal sheets. Actually, this is just for the shading purpose. All this body are built up with metal containers with airtight condition. They have also insulator in between. So commercially, you can build it from some authorized companies, through authorized companies, and inside that you will have a lot of sophisticated things to maintain the negative pressure and also to contain any chance of virus spreading and other things. This is the new ABSL-3 facility that we built here at ILS. This is the old one from top, you can see, and this is the new one, which has got all the earlier work we did with this one. The old facility was actually for BSL-3 related studies. It was a small chamber or room for animal related studies. So we had a capacity that time to handle only 30 to 40 animals in this BSL-3 facility. But this new ABSL-3 facility, we have now capacity to handle at a time around 300 to 400 animals. And you can see that the inside, this is the new facility. Different in both the sides, we have separate zones for large animals like hamster and also small animals like mice and other things. So we have already related to this facility. We have already displayed like what kind of services that we are providing here. What are this is the screenshot of the web page of our ABSL facility. It is publicly available in an ILS web page. So if you go there, we have detailed mention like what are the models that we have, what kind of services we are giving, what are the charges also that we are asking for different studies. And also how you can approach us, what are the forms that you have to fill, and who is the contact person. Everything is mentioned in this facility. So these are the this is a screenshot from the CCTV footage that we have for the different chambers or rooms that we have inside the BSL facility. So the micro city that I was talking about is placed inside the BSL facility. Probably, to best of my knowledge, this is the first ABSL facility in India which has a micro city scanner inside the facility. So because when we are dealing with infectious agent like SARS-CoV-2, we cannot just infect the animals and bring it outside for scanning. Everything has to happen inside the facility. So we have the ABSL facility equipped with micro city scanner, and also we got funded for this is our micro city scanner, and we have also funded to get more equipments which are essential for downstream studies like histopathology is a very essential thing when we work with animal models. So with the funding, we have a standalone facility for histopathology. We have automated microtome, tissue processor, embedding system. So, and also importantly, you need manpower. You need people who get trained and stay with you. So fortunately, we got some good people to work with us. And what I am talking today, actually, all this is because due to those people and my students, very hard and selfless work, actually. During COVID time, like when things were really very bad outside, these people didn't care. They have gone into it, and I'm very happy that so far we have already given services for 8 outsiders, companies like Davor and other people's candidates we have already evaluated, and we internally also have completed around 15 studies for our ILS facilities too. So all this thing happened because due to these people's involvement. So this is the form that we have displayed in our webpage also, like these are the different endpoint evaluation that we provide as service. And all details are available, and I'll just skip this. And this is so the charges initially that we have for pharmaceutical industries, academic startups. This is what we are giving for both antiviral and immunogenicity study. So this is what initially we have kept. So with this, actually, I have reached at the end. Thank you so much, all of you, for being patient and listening. And now I can take some questions. You can go for a discussion.
Yes, thank you, sir. Thank you very much. Now it's time for questionnaires. So any question from any corner can be asked one by one. If you have any queries, you can ask sir. Sir will answer that. Any questions, you can raise your hand. Now you can directly unmute yourself. Any questions from any part? You can directly unmute yourself and you can ask the question to our speaker.
And also, I will also like to mention that in addition to this animal challenge facility, at ILS, we have also two other facilities. One is antiviral candidate screening, in vitro screening of drug. That also we are providing on a service mode. Like before going to animal studies, if anybody has any lead candidate, they want to check in cell culture model whether they have antiviral efficacy or not. With a nominal price, we are also providing that service. It is also displayed in our webpage. Another new platform that we have added is the immunogenicity platform, which will be mostly for clinical sample analysis with people like companies who are interested to do antiviral clinical studies to check antiviral efficacy. Their serum samples and other things can be handled there. That was supported, that is supported by BIRAC. And the other antiviral candidate platform that you mentioned is supported by BBT. And all these three platforms are working in coherent, the antiviral, animal model, and immunogenicity platform. And definitely with fee for service. And if anybody is interested for a collaborative thing, that is a different, that all depends on the scientists who are involved, and all depends on how the things will be taken up. But mostly we are promoting a service mode.
Okay, sir. Thank you very much. Sir, I have one question. That is, if we want to go for any, there is tissue culture work or there is studies. So, how much amount of sample we have to send for screening, in specifically for COV2?
It all depends on what sample you have. Like if it is a crude ester, plant crude ester, like the concentration, what you have, what is the vehicle DMSO or water ester, or if it is a synthetic compound, like what is the concentration, the stock concentration that you have, because candidate to candidate, your amount, concentration, all this will vary. So we, before going for in vitro drug screening, first step is we check the cytotoxic effect, because we have to take a concentration of the drug which is not toxic to the cell where the virus will be propagated, particularly the virus cell. So first we in that service, when you do the antiviral experiment, first we give you the cytotoxic effect of this candidate. Then we suggest a concentration for antiviral study based on the cytotoxic profile.
Okay. Sir, at a time, how many samples can we screen?
We can screen 5-10 samples, not a problem. But even recently, we have got a high throughput drug screening platform. So you can even go more. So that's not a problem.
Okay. So any other questions from any corner?
Okay, I have one question. Good afternoon, sir. Thank you for a very informative talk. Am I audible?
Yes, please. Thank you.
Sir, actually, as in the slides, you have mentioned different animal models like hamsters, mouse, ferret, non-human primates, and you have been working with hamster models. Sir, my question is, sir, since the clinical manifestation when these models, when we try to infect them with SARS-CoV-2, so we get different clinical manifestations. So from there, how can which model mimics the human model very much? Like as you told, non-human primates, the manifestation that we get, that is very much similar to human models. Then sir, why do we go for hamster or mouse models? Like how much similarity we get in case of humans?
Right. See, similarity, when we talk about the similarity, the similarity, first thing is the degree of damage or manifestation, the organs which are involved, then the mode of damage. All these things are there. Like first thing is whether the organ, the lung, is getting affected or not. First, the second thing is the lung damage. Is it happening only in the upper respiratory tract or both upper and lower respiratory tract? And whether it is happening only in the bronchi or in the alveoli, all these things are important. I agree there will be, as I mentioned, there will be a difference between animal models and human. But the good thing is, at least we know that only after infection, the SARS-CoV-2 virus went into the hamster lung. It is infecting hamster lung cells, as we can clearly see that the viral proteins are present inside the bronchial cells and alveolar cells. And at the same time, we also saw that as this infection leads to recruitment of immune cells and blood vessels are also getting damaged. So which is mimicking most of the things that we see in human. But the extent, the duration of the lung damage, the extent of lung damage, all these things may be different, but the basic pathogenesis that the organ got involved and the tissues are also up to some extent getting similarly involved is good enough, at least to carry out some of the studies like antiviral study or a vaccine study. Because in antiviral study, what we see, a antiviral candidate either will stop the replication of the virus or will stop entry of the virus to the cells or will stop the unwanted immune effect that is aggravating the pathogenesis, right? So all these things can be evaluated in these animal models because all these things are happening there. But definitely, like before reaching to human ones, a lot of things, not only whether they are efficacious in controlling the virus entry or replication or immune system. In addition to that, another important point will be whether they are toxic or not, right? So that's how not only with COVID research, with any kind of biological research that we do with animal model, this question is always there: how much it will be effective in human ones? But you have to start with something, and nothing can be better than the intermediate one, which is animal model.
Okay, thank you, sir. Rightly said. Sir, somewhere I had read, sir, the coronavirus that affect these pangolins, it is very much similar to SARS-CoV-2. So, do pangolins are ever used as these intermediary host or as animal models?
No, to best of my knowledge, nobody has done that pangolin infection because they are endangered species. So nobody will ever ethically approve to use them as a model for study. Yeah.
Okay. Thank you, sir. Thank you very much.
Sure. Thank you.
Sir, I have another one question last. Sir, can we study that is 3C like main protease of SARS-CoV-2? This enzyme inhibition study, can we do it here, sir?
Yes. That also some assays have been developed. My colleagues, Dr. Soma Chaturvedya, to check the protease inhibitor study also in culture, in cell culture model. That assay system also they have developed.
Okay. Because sometimes we design some ligands.
Right.
We have actually in one project we have designed 4 molecules. We think it's probability that they may inhibit 3CL pro. So if required, we can also study there. That is the activity enzyme inhibition study. If you want to do.
Sure. It's possible.
Okay. Thank you, sir. So any other question from any corner? If there is any question, then we will go ahead, or we will call it a day.
Hello.
Any other questions?
Hello.
Yes, please. Yeah, just increase your voice a little bit.
Actually, this is a program of BPUT Odisha and for faculty development program. So you have, please give advice to the faculties so that they can be encouraged to go ahead with their research works and because so most of the faculties, they don't know the facilities available nearby and they do not dare to take the facilities available and conduct researches on bilateral basis.
Right, right. Thank you very much. Yeah. Thank you very much for asking this question. Actually, I'm glad that you raised this one. Actually, that is one of the reason of doing this kind of training. And let me tell you that now government is very serious. You must be reading in newspapers, and the government is very serious of sharing the infrastructure of research institute with other institutes. So every institutes are now bound to share their infrastructure and facilities to anybody who wants to carry out research. So the only condition is some nominal charges will be there, and there are also some other ways how to you can talk about all the authorities or scientists, and you can see if it can be done in a collaborative mode. But every institute, irrespective of the state government or central government, they are bound to share their infrastructure. And government is promoting now. Even the minister told that if any institute will show that they are sharing their resources with the other institute, they will be rewarded more. So it is a high time, like there is nothing called due to infrastructure, any idea should be stopped. So please feel free to contact any scientist at ILS, or definitely they will be happy to take your request. And you can even directly approach the director, ILS, no problem. He can forward your request to the concerned scientist in charge, and always welcome, and we'll be happy to take your request. And always, for my personal feeling is everything starts with a personal discussion with the faculty that actually facilitates the things better, rather than just coming through the director, because ultimately some scientist at the lab has to be involved. And if you have only one, two things to take care, like suppose you have already done a couple of studies, you have to do a confocal imaging to conclude something, you can come and direct. We have all the facilities, have also technical staffs or designated technical staffs. You have to just talk to them. They will tell that, okay, 1,000 per sample. You give your samples and you will get the results within stipulated time. So if that kind of things are also fine, but if you want to start something from the scratch, it is better to get involved with some lab, go for a discussion, and then take the things further. And who knows, it may lead to a collaborative research proposal in future also. So please feel to do the same thing, not only with ILS, with any institute around you. Please feel free.
Thank you very much.
Any other questions?
Sir, I have one more question. Sir,
Yes.
since you are using this hamster model, sir, how did you create that model expressing that human ACE2, or did you do something else?
Right.
Sir,
So,
yeah.
right. Sorry, probably I didn't clarify that one. So the
Was
point
it
is,
CRISPR or some other technology?
no, hamster by itself, nothing. These hamsters are not genetically modified. These are wild type hamster. Their wild type ACE2 protein is good enough to bind to this virus. So that is beauty of this animal. They are not genetically modified. They are wild type. You can even purchase from any vendor outside. And if you have a facility, you can do that. So they are not specialized hamsters. They are simple hamsters, wild type hamsters.
So then the transgenic mouse that you mentioned in that table, so those mouse, they were developed by this CRISPR-Cas9 technology?
There are two types of mice.
Huh.
The CRISPR-Cas is the technology people have used to generate a humanized ACE2 mice.
Yes,
Humanized ACE2 mice means, like even in the ACE2 mice, what Jackson lab is selling, that is a transgenic ACE2 mice where ACE2, the mice has also its own ACE2, because right, and in the mice ACE2 background, these mice are expressing additional protein, which is a human ACE2. But some people have, what they have done recently, they have deleted the mouse ACE2 genome. In that place, they have inserted human ACE2, which is called as a knock-in model. Knock-in.
Yes, sir.
So that people have done through a CRISPR technique method. Otherwise, the other ACE2 method, transgene method, is a conventional one.
Okay, sir. Thank you, sir.
Any other questions from anyone? I think we are not expecting any more questions. So,
Sure.
Arshida sir. Hello, sir. Hello, Arshida sir. Sir, can you hear me?
Yes, yes, I can hear you.
Sir, I think we don't have any more questions. So let us call it a day. Shall we terminate?
Please present vote.
Okay. So I thank Dr. Senapati sir. So you're so kind that you have given your precious 2 hours time, more than that, to us. And it was really insightful. We have got a tremendous knowledge. And I think our faculty members, our colleagues will be surely utilizing that not only in purpose or in perspective of COVID, because other cell lines are also you are maintaining. So any sort of biological work can be done here. So there is we got really enriched by learning these things. So I thank you very much, and we will wish that in future also you help us in the same way that you did today. Thank you very much, sir. Thank you.
Thank you. Thank you all. Very
Thank
nice.
Just,
Let
Thank
us
you so much.
Okay, sir.
just one thing I want to convey to the members present here,
Sir.
that the scientists of ILS are very busy persons, even though they are very busy, but
Yeah.
their response is very prompt.
Right.
Not expect a deny from them, and as soon as you ask anything, they reply immediately and they act immediately. So anybody or any institute they want to interact with ILS, I think they will cooperate.
Yes.
That I have remarked here.
Sir, sir, one request to you. Can I share your email ID if anyone asks me about?
Yeah, please.
Sure, sir.
Please.
Okay, thank you. Thank you.
Thank you all.
Thank you, everyone.
Thank
So
you,
let
sir.
us call it a day. Thank you, sir.
Thanks.